Okay. I think we can start now. So I think if there's anyone left to join, I think they will join in a couple of minutes, and they can always ask us during the seminar or later on the day maybe. So I guess we can start now. So my name is Enrique, and we have our colleague Andonis. We are both going to present you a little bit about this project, which is called NEOSUCCESS. And as some of you already know, it's about a simultaneous biogas upgrading and succinic production. So both things at the same time, both processes together. And today we're going to explain a little bit on about the project, about us, and so on. So as an outline, first, I'm going to try to introduce you to the project, to the consortium members, to the schedule that we followed, and then I will go a little bit on describing the process itself, this simultaneous biogas upgrade and succinic production that I was mentioning. And yeah, kind of the difficulties, the paths that we followed. And last but not least, Andonis is going to explain us some details regarding technical aspects on the unit or the design or the tests that we did, experiments we realized we did. So first of all, as for the project, it's a European project of research and development. So it counts with some European fundings from the Horizon 2020 program and the Fast Track to Innovation as well. And its goal is to upscale a biotechnological process that regards biogas and succinic. So basically what we wanted to do was studying this process in the lab scale and trying to think of a way in which we could upscale it into an industrial unit. So basically going from around 4 liters of fermenter to almost 4,000 liters. You can see that the dimension changes a lot, and that means a lot of challenges that we had to go through, but I think we did a good job. As for the consortium, it's 4 members. You can see where we are at. It's 2 engineering companies, both of them from Spain, one from the northwest and one from the east. And the role of the engineering companies was to think of a way in which we could come into an industrial reality, the process that was discovered in the lab scale. We also have another 2 members, which are the University of Thessaloniki and the Technical University of Denmark. And they were kind of the ones that gave the know-how and all the experience they had on succinic and on the process itself. Today, I am representing IVEM, and Andonis is representing the University of Thessaloniki. As for the facilities and where we installed the unit, it's in Paterna. So if you're not familiarized with the area, this is the city of Valencia, and our facilities are right here. So it's kind of 10 kilometers away from the capital of the region. Inside our facilities, where IVEM is managing a wastewater treatment plant, we have a specific area, which is this one here, that we often use for research and development projects, right? And in this case, here's when we start the upstream and the downstream. This would be the upstream, 2 of the fermenters, and this would be the downstream. As for the timing and the schedule, the project was, before it even started, it was planned to have different stages. So the first one, when the project started in June 2020, was basically designing and building the unit. We have to figure out how to come to that industrial reality, the process and the unit that they kind of discovered, right, in the laboratories. So once we had the unit built, the next stage was to validate the unit. That's basically testing it and checking that everything works properly, that there are no leakages, basically that the unit is ready to work. So kind of 2 years after starting in June 2022, the industrial unit was ready already to start operating. So the next step was basically, like I like to say it, playing with it. We had to operate with it, try different conditions, and of course, analyzing the results and seeing what was the best condition, how we improve, what can we optimize, what are the biggest challenges inside the operation, and so on. So basically, a couple of months ago, in July 2023, that's when we finished this stage, running a total of 10 different batches. And that's when we could say that the unit and the process were tested for the first time in industrial scale. As I said, we already did some tests in laboratory scale, even in pilot scale, that Antonis will mention later. But this was the first time that an industrial unit was built and operated with this process. And now we're in the last stage, which would be promotion, which is what we're trying to do right now, basically spreading a little bit the word, telling everybody what the work we've done and trying to promote a little bit the technology and the project itself. And soon, in November 2023, the project will end. So going to the process itself, which is the key point here, of course, how does the NEOSUCCESS process work? So first, we have two inputs. One is a gas stream, which is basically biogas, mostly methane and also CO2, that we put inside the fermenters. Then another input is a sugar-rich substrate that in our case comes from an industrial candy waste that we also put together in the fermenter. This is a liquid stream in this case. So basically, thanks to our tiny hero or main character here, which is Actinobacillus, we managed to ferment the sugar inside and produce succinic, eliminating the CO2 from the biogas. So we take out the CO2, leaving basically biomethane, and we transform this into succinic. So the following step, once we have finished with the fermentation, we're left with a broth of a mix of things. Some of the biomass coming from the Actinobacillus, some of the sugar waste that was not completely degraded or transformed into succinic, and we also have some other acids such as acetic acid, for example, or some other compounds inside the broth. So what we want to do is eliminate everything that we're not interested in and leaving the succinic acid as pure as possible, leaving as a result like a whitish powder that you will see in some pictures. So as I mentioned before, there are two subunits or two, let's say, parts of the process. The first of all is the upstream process, which is the fermentation and somehow the heart of the process. That's where we eliminate the CO2 and we produce the succinic acid. But as I mentioned, we have a dirty broth, if we can say it that way, and that's where downstream process comes in, which is basically eliminating all those compounds that we do not want and just leaving the succinic acid as pure as possible. So NEOSUCCESS also has two main products that I mentioned. One is biomethane and one is succinic acid. This is, of course, a gas, and this is a powder. So if you're not familiarized with these products, biosuccinic acid is a basal industrial compound that is used in many industries, such as cosmetics or bioplastics or pharmaceutical industry, food industry. I like to think it this way. If we're talking about Lego toy, maybe this would be a very important piece that you can use to create anything you want. That's why it's used in so many industries. And biomethane, I think most of you already are familiarized with it. It's a bio-based fuel that can be used for many things, right? For transportation, you can put it back in the gas grid of the city. You can generate heat, you can generate electricity. Today, we're going to focus on biomethane. Tomorrow, there's another seminar in which our colleagues are going to put the focus on the succinic acid. So if you want to have more information about succinic, I suggest you assist tomorrow as well to the seminar. As for the biogas, we take, in our case, in the unit that we built, we take the biogas produced in the wastewater treatment plant, and we make it run through the fermenter. Here, we have a device that allows us to count how much volume we have treated, and we have gone through the fermenter and transforming it to biomethane. Then that biomethane that comes out, we store it in a gas bag that we had to install in the facilities, and that allows us a bit of a buffer to return it back to the wastewater treatment plant. As for the biogas, the composition is mainly methane, which is the part we're interested in. There's also some CO2 and some other traces of steam and so on. The biogas mostly comes from some anaerobic processes, digestion of some organic matter. In our case, it comes from wastewater treatment plant sludge, but it can also come from the organic part of the urban waste, for example, or some other organic residues as well, such as residues from farms, for example. And the interesting part, as I said, is to eliminate everything and just remain with the biomethane. So there are many methods for this upgrading. You can have some water scrubbing, you can have some pH adjustment to eliminate the CO2, some activated carbon membrane. But in our case, NEOSUCCESS would be a biological upgrade, since this actinobacillus somehow breathes or eliminates the CO2 and eats the sugar to transform it into succinic. In the industrial unit that we built, the biogas comes from the wastewater treatment plant, either from the gas bag or from the anaerobic digester. This is the wastewater treatment plant of Paterna, and that over there would be the area where we installed it. The biogas has a mix of mostly methane and CO2 that we pass through the upstream, through these big fermenters. And thanks to actinobacillus, we managed to eliminate the CO2 and leave only the biomethane. In our case, inside the facilities of the wastewater treatment plant, we have some engines that allow us to produce electricity from this biomethane, right? So we take biomethane, we put it through these microturbines, and we generate electricity. Here you can see the microturbines. That's the anaerobic digester. And there on the background, you can see the facilities. Actually, this fermenter right here would be over there. So as you can see, it's pretty close. We generate the biomethane here, we pump it all the way to the microturbines, and we generate electricity for inside the plant. So guys, this is basically the introduction for the project. Now my colleague Andonis is going to go into detail on the aspects of the bacteria, on the biogas itself, and many other things. If you have any question, you're more than welcome to ask them when we finish the seminar at the end. We'll have some time for that. And now I'll give the turn to Andonis.
Hello, everybody. My name is Antonis Lithourgidis, and I'm a PhD researcher in the School of Agriculture at AUTH University, which is a partner in the current project. Enrique, can you stop the share screen so that I can share my screen?
Yeah, there you go.
Can everybody see my screen? Can you see my screen?
Yes, we can see.
Okay, guys, first of all, regarding the process that Enrique described, let me just first tell you that this is sugar-rich industrial conjugates is not the only substrate that it can be used. It's a general process, and we can produce succinic acid for a wide variety of substrates. Some of them can be dry pulps, corn fibers, palm tree bunches, leftover wheat flour, and of course, we notice around a lot of food and bakery waste, and also our own kitchen waste that we produce every day. These are all valuable substrates for the production of succinic acid. Another last one, very promising substrate is the algae. As we have seen in the last years, it's used for a wide variety of operations. But usually what happens with most of these substrates is that they need a pretreatment, either physical or chemical. So what does that do? It adds cost to our process. Therefore, we use the industrial conjugates, which have a very rich sugar composition, and we usually don't have to pretreat it to get good results. So in a general concept, the process design and development to produce succinic acid is usually we select a feedstock and we either go through some pretreatments with some enzymes or such, and then we develop our strain, in this case the Actinobacillus. Then we optimize our medium culture and we implement some process engineering, and then we are ready to validate and scale up our process. Of course, the final broth, eventually we have to purify it to recover the succinic acid so that it can be used as a chemical platform. This on the left, you can see some information about the theoretical stoichiometry and the CO2 fixation. 1 mole of carbon dioxide equals to 1 mole of succinic acid, which translate into quantities of 1 kilogram of succinic CO2 translates into 2.68 kilograms of succinic acid. So if our feedstock is only constituted of the purest form of sugars, the glucose, it can actually 1 gram of glucose can provide 1.12 of grams of succinic acid. Although we know the feedstocks are usually compiled of different sugars, and this is not possible because there are disaccharides inside. Okay, so all this process development, we have developed it with the industrial conjugates in our case, and for which we will talk later on. Regarding our macroorganism, it's the Actinobacillus succinogenes. This is his main fermentation route. So the sugars more or less get degraded and get consumed over time. And here we have a simplified metabolic pathway. After the node, the PEP carboxygenase, from one of the pathways, we have the succinic acid as the last major project. But for this to occur, we have to provide an inorganic carbon source to the system. So it chooses that pathway. The inorganic carbon source can usually be provided as different salts or in gaseous form of CO2. So what does have CO2 inside? It is the biogas actually, and that's how we achieve the biogas upgrading. Therefore, to provide a certain amount of inorganic carbon is a key contributor to the process. So why do we actually study the Actinobacillus succinogenes? Of course, there are other candidates with near similar performances, like either engineered or not, and some yeasts. But Actinobacillus succinogenes, based on our previous experience, is proven to be a very robust microorganism, and it can actually utilize a wide range of different feedstocks and sugars and behave a bit better during substrate and product inhibition phenomena. Let's go over the process in a general sense. We have inside our substrate, in this case, the industrial sugar-rich waste, and then we have an inorganic carbon source, the CO2 from the biogas. Both of them go inside the fermentation with our microorganism, and then we have the two final products, as also Enrique told you, the biomethane and the succinic acid. If we want to scale up this process, we have to perform consecutive fermentations, and afterwards we have actually to treat the succinic acid to use it as a final product to separate and purify it. Usually it consists of a series of processes like centrifugation to remove the biomass, activated carbon, and microfiltration to remove any pigments that interfere for the next purification steps, ultrafiltration to remove any remaining solids and any other big compounds such as proteins. If you have ever seen a membrane clog, you can see a big jealous a big jealous compound on top of it that usually represents the proteins. And afterwards, we proceed to the nanofiltration. There, usually we eliminate the sugars in the broth and other main co-products, although sometimes nanofiltration cannot be that much effective for the byproducts, for the acid separation. That's why we have the next steps, vacuum distillation, where the byproducts get evaporated, and then we can crystallize our final product, which is liquid succinic acid. We just can bring it to a solid form. Instead of vacuum distillation and crystallization, we can apply the spray drying, which actually does the same thing simultaneously, evaporates the other acids while keeping inside the succinic and make it into a powder compound. Okay, now let's focus on in the next slides on the fermentation part and on the biomethane and the succinic acid production. For those that have not ever seen a reactor design, here I present you a general concept of the reactor design for a fermentation. It doesn't matter the scale, usually the logic is the same. And here is their characteristics. Here we have the agitation, of course. We have multiple sensor ports to monitor our process like pH, temperature, carbon dioxide, oxygens, and others. Of course, we need temperature regulation during the process. Here you can see a double outer wall sealing of steam where you can cool off or heat up. But usually in lab scale, we use electrothermal jackets to keep the temperature under control. Of course, a very important system is the gas sparding system, where a sterile gas is coming through inside the fermenter. It starts sparding inside the reactor. In our case, it's carbon dioxide. It's through the biogas. Therefore, we sparge biogas inside the fermenter. It's very important to use some filters before, so that doesn't the biogas contaminate our broth. In addition, we have multiple ports like sampling ports that use acid and base through pumps to regulate the pH during the process, and some additional ports if we need to add some nutrients during the process. Some additional features is that all this monitoring happens through a control system, a control unit that the company provides depending on the reactor. And we can see and monitor from our computer through an interface. Some reactors usually have a sterilization ability, which does not happen usually in a lab scale. Let's see a few more stuff about the procedure. Usually, this is the procedure that the recipe for the medium, culture medium that we have concluded to be the best in our case. For the first inoculum, usually we use a combination of chemicals, and then we use a salt, carbon MgCO3. And for the inorganic carbon salt, the first time in small bottles we use glucose. It's more operatable. And therefore, for the next fermentations, this is the composition you can see where we use biogas, and there is the CO2 for the inorganic carbon source, and we use industrial candy waste. Let me show you here a typical growth in a batch mode system in a pure culture. We start the process, and our microorganism usually has some lag phase in the start, then it starts growing really fast in the exponential phase. Then we have for some hours the stationary phase, and then the death phase follows. The times are relative, don't pay much attention to it. And here I also attach a typical profile fermentation in batch mode. You can see how the fermentation, when it starts, then we see the sugars getting consumed and the acids being produced. Of course, every time we will have some byproducts except the succinic acid. It's almost impossible to avoid them. But what we can do is to optimize the process so that we have better ratios of succinic to the other byproducts. So usually what happens, we use 5 to 10% inoculum. Every fermentation, we started as 5%, but we concluded that 10% is the best case scenario that provides a more strong fermentation in the next steps. So what does that mean? A fermentation will represent the 10% for the next fermentation, and therefore, depending on the volume that we want to reach, we do similar fermentations. So usually what happens for the first stage, we only use a big lag phase until we create the first inoculum. It takes 10 to 16 hours. We need the inoculum to take it in the exponential phase. And for the intermediate steps in the medium fermentations like small scale fermenter and pilot fermenter, usually takes 2 to 5 hours. for it to reach the exponential phase, and then we are ready to inoculate our final fermenter. Of course, I understand 10 to 16 hours, 2 to 5 hours. It's not possible in a workday if someone or his colleagues that work 24 hours. So if the logistics are not feasible, its fermentation step can last up to 24 hours. But the next fermentation will just appear with a bigger lag phase, but they will still work. Of course, the size of the operation depends on the application that we want, the available substrate we have, the chemicals and the equipment. And in this case, let me remind you again that is a pure culture fermentation. So sterilization is the most important aspect. If we have at any point of the fermentation and contamination, then we just disrupt the whole process. Of course, except the batch mode, there are other systems like continuous system fed batch, where during while the fermentation works, for example, in the stationary phase, we can add some extra substrate or some fresh inoculum, and we can prolong the process longer. Let's move on and see some of the protocols and the setups. I know this will be a bit much for you, but then I will show everything in pictures so that everyone have a visual. We start with inoculum bottles. It's small lab bottles. We weigh the chemicals, we prepare our media, we seal our bottles, we flush them with nitrogen to ensure anaerobic conditions. We then sterilize, and then we are ready to add our organic carbon source with a filter, of course, not to contaminate our process. We adjust our pH, and then we are ready to inoculate and let our microorganism grow inside the saging incubator. Therefore, we can use these to move on to the bench scale bioreactors. After we have prepared all our preparations, we add our medium inside the fermenter and some antifoam so that we don't have any problems with the tubes or the sensors. We calibrate our sensors, we seal our bioreactors, we put them as a whole inside the autoclave. Then we add our organic carbon source, either it's a glucose through filter sterilization or industrial candy waste. At this point, usually we pasteurize separately the industrial waste, and then we add it before initiating the procedure. After everything is inside, we sparge with nitrogen to ensure the anaerobic conditions, and we set our parameters, pH and temperature, to our optimal ones for our microorganism, and then we are ready to inoculate and go on and monitor our process. Of course, during the pilot application, most things are the same, but here it becomes at some points a bit easier, because what we can actually do to save time and costs is we can add all the chemicals and the substrate straight away into the fermenter before sterilizing. So there we can actually pasteurize or sterilize. In this case, we did pasteurization because the industrial candy waste, when they get above 100 degrees, usually they caramelize, and then we degrade the sugars before even the fermentation starts. Then we continue normal procedure like nitrogen sparging, set our parameters, pH and temperature, opening up our stirrer. Then we can actually start inoculating, and of course initiate our biogas sparging to allow the Actinobacillus succinogene to go through the C4 pathway to actually bind the CO2 and produce the succinic acid. Of course, its system, I understand, has its own small variations, but the general concept remains the same. Once someone understands the procedure, he can adjust it so that he does what he does best, how he knows to achieve the optimal result. All these have some dependability on the experience of the person, the preferability, the equipment, and the availability. So now let's show you some pictures, because the picture equals to 1,000 words. So now we have our microplates. Where is the microorganism? We weigh our chemicals, we prepare our culture medium, and then after sterilization, we are ready to insert our microorganisms inside in a saging incubator, where the microorganism will grow healthy. And then we can take that as inoculum to prepare more bottles, pure culture bottles with our microorganism, which can be used for our fermentation and inoculation. In the meantime, we can actually prepare our substrate. Usually industrial candy waste is very condensed, and we need to apply some kind of dilution before using it. We put it in an oven to pasteurize it as high as we can, but not reach sterilization because they will caramelize. And then we are ready to add inside our substrate in the fermenter, and eventually we can actually also inoculate it and start the biogas sparging. When the process ends, we can either store our broth for further analysis, or we can use it for further inoculum to actually upscale the process. So let's see some of the systems that we have used during the project. This is a general representation of a lab scale system. You can see the bioreactor, you can see the biogas gas bag through where the biogas goes inside the fermenter through a stereo filter from a peristaltic pump, and sometimes we can use pressure gauge to actually increase the headspace inside the fermenter to optimize the process. We have also hooked some acid and base control to control the pH, and CO2 sensors, which all lead to control unit and our interface in our computer where we can evaluate the whole process. Other setups that we have used during the project is a continuous biogas sparging. So the biogas just comes in from the bottom and goes out of the top. There is no recirculation, and pressurized continuous biogas sparging. Happens the same. The biogas comes from the bottom and comes out of the top, but there we apply some pressure on the headspace. And the C scenario is the gas recycling. It's the same amount of biogas goes around and around in loops until the whole carbon dioxide is consumed and we have biomethane from our process. In these cases, we have achieved most times over 90% of CH4 methane purities, and the maximum purity that was achieved was over 90-80%, which is quite an outstanding result, and it can be injected directly to the gas grid. Let's see now a bit of this process at the pilot scale. How we prepare. We have set up our base and acid bottles. Usually we need big quantities in pilot scale. We open up our fermenter, we clean it, then we await our chemicals and add it together with the substrate, and then we initiate the pasteurization. Then we are ready to actually inoculate and control our system through the SCADA system. We have automatic valves. We can set parameters and just keep an overview of the process, and then we just work. We let it work. We initiate the biogas sparging, of course, and we let the process work to ferment for 24 or 48 hours, depending on what we wish to achieve. And afterwards, we are ready. After we have collected both the succinic acid broth and the biogas in bags, then we can move on with our two valuable products. Okay, all this, now that I have described the process, now I want you to see how does all this actually translate to a real industrial unit. More or less, the protocol is the same. What happens here is because in industrial environment, it's harder to keep a fermentation pure. The most important step is the cleaning and the sterilization of all the components. We usually apply a 3-step sterilization. We pass water, a strong base, and some steam to clean everything. And then here we have two options to prepare our culture media and our substrate, either separately in storage tanks and pasteurize them or sterilize them there, or we can pump them inside the reactor and do the whole sterilization part there. Of course, we need to hook up our sensors to calibrate them to ensure anaerobic conditions and set our parameters at the set optimal conditions. And then we are ready more or less to inoculate it, to inoculate our fermenter and start the process by initiating also the biogas sparging. In this case, we would need some very necessary components. The most necessary is the steam generator, which actually saves the whole process. We have chillers, we have decalcifiers, we have a big storage tanks for acid and base needed during the process. We have nitrogen tanks that can be used to ensure anaerobic conditions. And of course, we have a laminar flux and extractor hood where the first inoculum coming out of the lab will transfer it through there under sterile conditions to our fermenter. As you understood from until now, the sterilization part is the most important part, either at the start or either during the process. If contamination happens, it can disrupt the whole process. So let's see all the process in pictures. So the candy waste are being carried. We keep it inside the storage tank. In the second tank, we will prepare our culture medium. And afterwards, we are ready to pump them all in the first fermenter, the pre-inoculum fermenter, which we say. There, we actually initiate the sterilization, and then we are ready to to start the biogas sparging and the inoculation to let our fermentation work. When after that happens, after 24 hours and the inoculum grew, then we are ready to move it through pumps and pipes to the next fermenter. And after that, the same we will replicate it in a bigger scale, as Enrique showed you, in around 4,000 liters fermenter, where it also has the ability to actually recirculate the biogas. So over time, it gets upgraded more and more, and we get better methane qualities at the end. As Enrique showed you, this is a gas pack that we can collect and either burn it usually in situ or transfer it to the gas to the grid system. And there is also the storage tanks that we can keep our fermentation broth until we purify it. Some of the important parameters, components is our SCADA, our control system. Usually we have an overview of the whole process, each fermenter separately, and every valve we can set it automatic or not. Behind the fermenters, we have some extra panels that we can use to check in situ the pH and note it from inside the control SCADA system. It's very important to check the condition of the flow meters and the biogas pipelines that it comes from the plant. It's very important to see all our pumps for beta and acid regulation to be all in fine condition, and our pressure reduction for the biogas coming from the unit. Regarding the biogas filters inside the fermenter, I would say is one of the most important parts during our fermentation. We need to always check they are in good condition, because from when the biogas is coming from the biogas plant, it usually carries around microbes which can actually contaminate our process, and then everything will come to an end. It's very important to keep it in a good condition, and that we have tested that they actually are efficient. So let's do a synopsis on the fermentation part. We have tried different things over the time, the course of this project. We have tried to increase pressures to actually increase the CO2 binding and the succinic acid production. We have tried different recipes to increase the inoculum strengths. We have also tried microorganism adaptation, where actually the microorganism gets adapted over time to higher sugar concentrations, and it can behave in a better sense. There is also the ability generally to apply some genetic engineering and some pretreatment, which in the case of our candy waste, we didn't have to actually do it. So what is our achievements? In most cases, we actually managed from a typical biogas composition, 60% methane, 40% carbon dioxide, we managed to improve it to 90% methane, and our maximum value was 98%. That also translates to 90% of CO2 removal. And let's see what we achieved regarding succinic acid. In lab scale, in small bottles with an adapted microorganism, we achieved around 80 grams per liter of succinic acid, which is an outstanding value. And in a more pragmatic and realistic application in the pilot, we achieved around 40 grams per liter, which is quite much for such application. So regarding the challenges, so every process, you know, when you try to evaluate something, if it works or not, it will present challenges. The first and most important challenge in this process is the sterilization. If we cannot apply sterilization, then we just contaminate everything and we cannot work. The next things that we need to pay attention to is the substrate and product inhibition, which usually can be overcome with some microorganism adaptation. Also, the high amounts of base that we require to regulate the pH in the optimal conditions for the microorganism. Many people usually don't pay attention to this, but in my opinion, it is not negligible, because when the acids are being produced and when we actually also saturate the liquid with CO2, the pH is dropping down very rapidly, and we need to keep track of it and to pump it right up. And in an overall sense, we have to have an effective engineering after all this testing, so which allows to the minimization of effort from the staff, and so that we can easily maintain it if we need to fix something over time. I will not keep you any longer. I will just tell you like a few things about the purification. During the project, we have tried different purification methods. I will just present two of them. So our most developed one and the base case scenario. One of the methods is a pre-purification is common, first of all, for both methods, to include centrifugation and activated carbon treatment and ultrafiltration. And then we go to some ion exchange, nanofiltration, vacuum distillation, and crystallization. With this method, we actually achieved a very high purity of succinic acid, over 93%, which is quite valuable, and it can be used almost for all succinic acid applications as a chemical platform. And the recovery was about 85%. And the other base case scenario that we used just to compare it with our scenario is the two method where we didn't use ion exchange and nanofiltration. We went straight to acidification and then vacuum distillation, crystallization. The recovery, as you can see, was about the same. But what happened with the purity? It dropped very much, around 26 to 38%, which is usually not very preferable for any application afterwards. So let's see how the industrial unit based on this was actually built. This is a general diagram for the industrial unit. We have applied a centrifuge, a filtration, a pH control tank to reduce the pH before the nanofiltration, and then we have also a spray dryer to get our final product. You can see at the bottom the diagrammatic scheme, how everything is connected in a series, and we move from each step to the next and so on. Let's see all this in pictures. We have a centrifugal separator, alpha Laval, and then this has all the storage tanks for after each step, the liquid, the permeate goes inside the storage tank, and then for the next step, a pump sucks it out, goes through the filtration, and then puts it in the next tank. So here you can see in the ultrafiltration, it's to the small meshes. After the centrifugation, a working flow of around 80 liters per hour and the max working pressure around 10 bar. It's compiled of two mesh filters, 5 and 1 microns. And then we have a pH control tank where we can add acid so that we drop the pH so that the nanofiltration is even more effective. Here is the message from the ultrafiltration, and here the white column, you can see it's the next step, the nanofiltration. Regarding the nanofiltration, the max working pressure of this machine is around 31 bars. Usually nanofiltration is affected above 7 to 8 bars. And it has a quite big pH operation rates, T3 up to 10. Also, let me present to you the final step, the spray dryer, which is also, if you can see its shape, is almost like a silo. It has on top of it a centrifugal spray head where we set the frequency and the heat temperature inside the chamber, and then we spray our liquid from a nozzle, and then when we set the temperature above 140, for example, we evaporate the byproducts, acetic and formic acid, and then, but succinic acid is kept in there so that it dries out, because the succinic acid has higher boiling points over 200 degrees. And then we actually get our final product, the succinic acid powder, which is so much valuable and can be used for so many things. If someone wants to see more things and in depth, you can actually check some of our publications in the frame of this project. One of them refers to the pilot scale fermentation and the dowsing purification process. One of them refers to the CO2 profile and the optimization in the succinic acid production by applying different pressures in the fermentation. Another valuable publication is a wide range exploration of the potential of the industrial range under various setups. And also, we also have published a techno-economic analysis for the whole process for the succinic acid production from sugar-rich wastewater. Thank you all for your attention, and I hope it didn't take much time and effort for you to listen to me.
Thank you very much. Now, guys, if you have any questions or any comments, I don't know, any topics you'd like to discuss, maybe, you're more than welcome to ask us. You also have the links to our website, to our Twitter, to our LinkedIn. Can you show it again? Can you share it again, Andonis, please? So they can see the link.
Yes, let's give me a moment.
And you also have our
Can you see it?
Email. Yeah. So if you want to contact us, you can ask us now, or you can ask us tomorrow, or whenever you want to ask us again. So do you have any question, any of you?
If someone has any questions,
observations.
yes. Also, you're free to join our tomorrow's webinar, where two other people will also present some stuff of the project, and they will focus on different parts mostly. And here, if you think any question from today, you can actually also ask us tomorrow. We will be there. We would like also to tell you that this will be recorded and uploaded online. So if someone wants to come back to review any information or any links, he's always welcome to. And in case you have any questions, you can always contact us, as Enrique told us.
If there's no question or no observation you guys want to make, I'll close it for today then. And as Andoni said, tomorrow, same time, 12 or 1 p.m. in Greece. There's another seminar as well, and mainly focused on the succinic and downstream part. On Friday, we will have a live demonstration and a visit to the facilities here in Valencia. So you're always welcome to come. You have our contacts, and feel free to text us on WhatsApp or on the email or through LinkedIn or any other way. Okay, so thank you very much, guys, and that's it for today for the seminar.
Thank you everyone for attending.
Thank you very much.
Thank you. Goodbye.
Ciao. Ciao.